Good morning, everyone. It's 9:30 already, so we're supposed to start. Welcome to class number 7 of the course Deep Learning by Example on Beowulf. This class is a part of the series, but each class is supposed to be standalone. So for those of you who did not take previous classes, I will try to provide the background information which will be hopefully sufficient for you to understand the contents of this class. The topic of this class will be message passing and self-attention-based networks, data augmentation, transfer learning, and their application to drug molecule property prediction. This class is being recorded, so if you don't want your face to be in the recording, please make sure to turn off your video. I will send email to everyone when the recording is available on our website.
Yeah. The press, but I don't know whether it does not let off or secure.
One second. After presenting each slide, I will make a quick break, quick stop. Please unmute yourself, everyone. Please keep yourself muted. But after presenting each slide, I will allow a few minutes for quick questions. If you have a question, please unmute yourself and ask the question. Let's postpone longer questions and discussions till the end of the lecture when we are supposed to have some time. The entire class will last for 2.5 hours. During this time, we are expected to have 2 breaks after approximately each 50 minutes. So, and 5 minutes breaks. Any questions so far? No. Proceed to the first slide. In the area of application of deep learning to molecules, there are 2 fundamentally different approaches. In one approach, a molecule is represented as a graph, with nodes of the graph being the atoms and the edges or links of the graph being the chemical bonds. In another approach, the molecule is represented as a SMILES string, where SMILES stands for Simplified Molecular Input Line Entry System. Example is shown here. You can think of it basically as a chemical formula for the chemical substance, but it's not exactly true. While the graph representation of a molecule is unique, there is only one graph per molecule. The SMILES representation is non-unique. It is ambiguous, meaning that multiple different SMILES strings may represent the same molecule. And that multiplicity is actually a consequence of the fact that most of biologically active molecules are not linear chains. They are branched. And example is shown here. Here for this molecule, we have a backbone chloride, or say starting from nitrogen, carbon, 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 chloride. Carbons are not shown here, as well as hydrogen ions are not shown here. And there is a side chain. In this case, it's double bond O, oxygen. And in the SMILES string, the side chain is shown included in parentheses. However, there is alternative representation of this molecule. For example, you can regard this oxygen, carbon, nitrogen as a backbone and the rest of the molecule as a side chain. In this case, you will have this representation. Or you can think of oxygen carbon chloride as a backbone, and a single bond nitrogen is a side chain. In this case, you have the third representation. And you can think easily, imagine that the longer the molecule, the bigger the molecule, the more branches it has, the more different possible SMILES representations would be available, which correspond to the same molecule. Whatever SMILES representation is used, the next step would be to break the SMILES string into individual tokens, like shown here, example. And then the recurrent layers, deep learning layers, capable of processing sequence data or other similar layers, which we'll discuss later in this class, number of hydrogen ions connected to this heavy atom, and so on, hybridization and stuff like that. And the features possessed by the edges of the graph would be, for example, the type of chemical bond, which may be single, maybe double, maybe triple, or maybe aromatic. So in the so-called one-hot encoding, each of these features for the entire molecule can be represented by a matrix, a certain matrix of zeros and ones. In addition, for complete definition of a molecule as a graph, we also need adjacency matrix, which specifies which actually nodes or atoms are connected one to another. And that adjacency matrix, we discussed it in the previous class, is also some matrix of zeros and ones. Now, for processing the molecules represented by graphs, graph convolutional layers can be used, or similar layers. We will discuss another possibility in this later on in this class. There are a number of non-biological applications that exploit the techniques similar to those to be discussed in this class. One example is prediction of quantum mechanical properties of organic molecules. And the most up-to-date technique used in this area is called message passing. We will discuss it in details in this class. This message passing is actually generalization of the graph convolution transformational layer, which we discussed in the previous class, and I will refresh your knowledge about the graph convolution later on. The other example of application is natural language processing, which deals with analysis of text documents. However, there is a pretty interesting analogy between processing the text document and processing a data set of SMILES strings. Between individual sentences in the text and individual SMILES strings in the data set, and between individual words in the sentence and SMILES tokens. And this analogy allows us to apply some techniques developed in natural language processing to analysis of molecules. The biological example of this class, I called it MP-SAN-MP, message passing and self-attention based networks for molecular property prediction, will make use of both graph representation and SMILES representation, or either one or another, and will allow us to exploit all of these techniques that I mentioned previously. I'm done with the first slide. Any questions so far? Okay, I proceed to the next one. If you later on have questions about this slide or any other previous slide, you are welcome to ask those questions in the after presenting next slide. So this class will be the last in the series, and let's see how the topic of this class is related to the topics of previous classes. First of all, this class, the approach of this class will be a supervised machine learning approach, which makes it similar to the approaches of classes 1, 2, and 6 but the topic of this class is, of course, very different. The second comment is that this class will data extended set of data which was used by class number 5. The class number 5 also was focused on drug molecules, but on design of drug molecules, not property prediction. And next comment is that in this class, we will discuss 2 new layers we have never discussed in previous classes. This is message passing layer or transformation and multi-head attention layer, which actually originated from natural language processing. And the final comment is that the central topic, or one of the central topics of this class, will be how we fight overfitting. And we will explore 2 approaches to fight overfitting. One is data augmentation, and the other is transfer learning. The data augmentation is basically generation multiple alternative smiles strings. This way we would effectively increase the size of a data set. But transfer learning is more sophisticated topic. We will discuss it later on. Any questions so far? No questions. Next slide. So let's now start with the first prototype example of this class, which will deal with cartoon molecules represented by graphs and will allow us to introduce the message passing network model. To this end, I will consider a cartoon representation of a molecule. The molecule will be a linear chain, unbranched chain of atoms, of say M atoms. On the picture, it's 7 atoms connected by links or bonds. Besides, each atom we need, in addition to, we need to specify which atoms are connected by links. This is done by adjacency matrix, which we discussed in previous class. But in the context of graphs applied to molecules, usually a non-sparse version of the adjacency matrix is used, which is called pair indices matrix, and it's shown here. Each couple of indices in this array or matrix corresponds to non-zero value of the corresponding column and row in the adjacency matrix. For example, this 0, 0 means that 0 column, 0 row has non-zero value here, and so on. I mentioned in the very first slide that the features assigned to the atoms and to the bonds of a molecule can be represented by some matrices of zeros and ones. Accordingly, for this cartoon example, I will make assumption that each node or atom possesses a vector of n binary features. On the chart, n is 3. And each link or bond possesses L binary features. It is, for example, 2 on the chart. And when generating the cartoon molecules for the data set for this example, each link and each node will be assigned a completely random set of features, binary features, zeros or ones, except probably a certain part of the molecule, which is called motif. It is shown in red color. The motif, for the motif, we will use fixed, very specific values, and it will be the same for every time, for every molecule. The motif may or may not be present in the molecule. If a motif is present in the molecule, then we will make assumption the molecule will be considered good for treating a hypothetical disease and will be assigned a ground truth label 1. Otherwise, the label will be assigned, the molecule will be assigned label 0, and will not be supposed to help in treating this hypothetical disease. And the task of this example is to train a message passing network model on this data so that it could predict the class labels for new, previously unseen cartoon molecules. I'm done with this slide. Any questions?
I have a question. I was wondering where the 1, 0 is in the pair indices matrix.
The 1 and 0, I mean, in the pair indices, it's a different. I'm talking zeros and 1. Oh, again.
Oh, sorry. I mean the 1, 0. You see, there's 0, 1, but 1, 0 in the symmetric position. Where is that in the pair indices matrix?
1, 0. I do not see 1, 0.
Because in the adjacency matrix, the row 1, column 0, there's a 1 in it, right?
Once again, I'll explain you the meaning of the pair indices matrix. So if you should take first, pair indices matrix is in the way it's a non-sparse 2 element in the pair indices matrix.
Oh, sorry. My question was just that I think it is missing one pair in the pair indices matrix, which is the pair 1, 0.
1, okay, 1, 0. Oh, you may be right. Let me. Maybe you're right. Let me just not do it right now, but maybe I will look at the break, look more carefully. Maybe I'm missing something. Maybe just mistyping something. But you understand the idea, right?
Yeah, I don't understand the idea.
If you see the error, it might be too many things to correct. I'm not sure, but I'll look more carefully when I have more time.
Okay, thank you.
Not immediately. Yes, of course, of course. Or I will respond to you if you send me email. You can. I'll respond to you later on after the class. Anyway, any other questions? Okay, I'll assume that so far no questions. But again, if you have the question about this slide or previous slide later on, we will. You can do it later. So this slide presents the algorithm for message passing transformation or layer, and it explains how this message passing transformation relates to the graph convolutional layer or transformation we discussed in the previous class, in class number 6. So let's start with refreshing our knowledge about the graph convolution. In brief, in the simplest version of graph convolution, which is called vanilla graph convolution, there are several flavors of graph convolution, but the vanilla graph convolution, basically what it does, it's also called filtering. And what it does is it performs in one step of update of features of all nodes of the graph, given for each node, given the values, the old values of features of this node and its adjacent neighbors, which we called one-hop neighbors. One-hop means the two nodes are one-hop neighbors if they are connected by exactly one link. And the node itself is also one-hop neighbor of itself because it has a self-link. So the idea, say, we have 3 nodes, adjacent nodes, A, B, C, with all the values of features, XA, XB, XC. And our purpose is to find to compute the new updated value of feature of the central node, which is designated YB and shown in the turquoise color. And the formula, the general formula for this update is shown here, is basically YB would be a weighted average of features of all the one-hop neighbors of the current node, where the W is weight, WI is a weight, adjustable weight, B is a bias, which is also adjustable weight, and summation is performed over all the one-hop neighbors of a current node. For node XB, node B, that would be A, B, and C, adjacent nodes, one-hop neighbors. And the dB here is the degree of the central node, which is in our case, it's number of one-hop neighbors, which in this case is 3. That's about the graph convolution. Now let's see the message passing. The message passing algorithm for performing the similar task of updating features of a node is performed in 2 steps rather than in 1 step. The steps are called message step and update step. And the message step is in a way similar to what we discussed for graph convolution. In formula is similar, but with 3 important differences. The first difference is that the result of this computation, the message called message, is not assigned immediately to the updated node like we did in the previous case, but it is stored in a separate intermediate variable called message. The second difference is that in the computation of the message, we do not only use the node features designated xi, but also we use edge features. This is lowercase e with indices, and on the chart, the edge features are schematically shown as rectangles. There were no edge features taken into account in the previous example in the graph convolution. And the third difference is that the message step interspersed with the update step can be iterated several times, not just one time like we did for graph convolution, but several times. The default number of iterations is 4, but you can use any number through command line options. Now about update step. The update step computes the final value of the feature that will be assigned to the new updated node. And this is performed using the update function. Update function may be different, and it depends on your choice. But in our implementation, the update function is GRU cell. It's basically the update function is a function of 2 arguments. The message is 1 argument, and the old value of feature of this node is the second argument. And in general, this function is not identity function. So the value of the new value of feature will not be the same as message. It will be different. That's basically it. So in conclusion, the message passing filtering, also can be called filtering, is performed in 2 steps, the message step and the update step. This interspersed steps are iterated t times, where t may be more than 1. The message computation involves both the adjacent features and node features and edge features. The conclusion that I did not mention, but it is true, the edge features are not updated during the message passing. The edge features are fixed, supposed to be fixed, and only the node features are updated. And overall, the overall general comment is that message passing filtering, just like graph convolution, is generalization of graph convolution. Actually, there will be some homework assignment which show you that this is true. You will be suggested to find a limiting case of message passing where it basically would be the same as graph convolution. And the final general comment is that message passing, like graph convolution, is a local transformation, meaning that the new value of a given node, updated value, will depend only on this node and its adjacent node, but not distant node, distant from the central node. Any questions?
Yes. I have a question about the iteration, because where is the feedback when you get updated to the YB? How do you, in the next round, what is updated in the X to predict the B and B? So the iteration, how if because it's an iteration, I would suppose it should be take some information from the previous round and then feed in the next round.
Right. So, yeah, it's again, maybe I was not clear when I mentioned that first. When you do first iteration, say one iteration, you update all the nodes. Now the new values of the nodes, updated values, will be now made old values, right? And then you do the same. Basically, now you regard those which were after the first iteration, they were this turquoise color nodes, they now become all blue color nodes. And you do the same exactly, the same operation. So you do not use the same value of the features XB, for example, at each iteration. They will be different every time. They will be updated. Does that answer your question?
So it's updated from YB, it gets some information from so you have XA to start with. And then after first iteration, the XA will change, right?
That's correct.
How it changed?
That's correct. It is changed basically say you have originally X, then you have a set of Ys, YA, YB, YC. Now you make Ys again X's.
Oh, okay. Thank
and
you.
Then that start from all over again from now. You've got it, right?
Yeah. Okay, thank you.
Okay.
I have a question. So how these update functions differ from what we got from backpropagation?
It's a little bit more sophisticated. Backpropagation applies to the entire network. I am discussing only one layer in the network. So it's kind of different. I would say it's different topics.
So what's the benefit of this from that? From backpropagation, how should we convince?
It's not benefit. It's because they can be used together. As I said, the message passing is just one layer in the network, and the backpropagation applies to the entire network with multiple layers. So these are two different entities, and you can use both. It's not like advantage of one over another.
But the accuracy is different, because backpropagation is depend on our loss function and how it can improve. But I guess I don't understand the value of this update function.
Okay. I think that this is not exactly the question about the current slide. So I suggested earlier that more general questions and discussions, postpone them till the end of the lecture. We want to be able to finish within our time of the class. So I suggest that we can talk about it later on, because it may be a longer discussion.
Okay.
Any other questions about this slide or previous slides? No. Then I proceed to the next slide, which presents a training code for the model that I mentioned previously on the first slide, the entire message passing network model. And the model schematically shown in the chart on the corner, upper left corner. The input to the network would be a graph represented by node features, edge features, peer indices, and one molecular indicator. I will comment on that one later on. Now, the entire model comprises several tensors. Tensors are shown as rectangles connected by layers or transformations between the tensors. The first layer is message passing, which we just discussed. There is also readout layer, which I did not discuss. It's kind of composite. It's not a regular Keras layer, but it is composite, comprising several regular layers, such as normalization or dense layer. And the purpose of readout is basically to do some kind of post-processing after message passing. And the last layer in the network is dense layer, which outputs a scalar. And that scalar would be actually the that scalar will contain the computed property value. In this case, it will be binary label, 0 or 1. And the entire code shown here, actually, the good news is that it represents, shows all the 4 major processes and steps that we discussed in previous classes that are found in any model we discussed in this course. This is the header, and getting data, defining the model, and running the model. The header part of the code in this case is a set of import statements followed by some definitions of the hyperparameters, some constants to be used by the code. The getting data part is basically getting the molecule. It is computed somewhere else because there's no room in this code and so it's a separate function. You can get synthetic data, and you get the entire graph corresponding to the synthetic molecule, but you can say modify on your own the motif that you will be using. This sample motif is shown here. By default, this sample motif will be used, but you can modify it as you want. Then the defining a model starts from this input of tensors. The tensor X is tensor of node features. E is a set of edge features. Pi pair indices, and the molecular index is a new item that we did not discuss previously. That molecular item is present here because the entire code is taken with minor changes from the biological example, where the molecules are not necessarily of the same size. For this example, we just use fixed M number of atoms in the molecule, so it will be the same, all the molecules will be of the same length. But for real molecules, they may have different size. And the molecular indicator, molecule indicator, basically, you see, it indicates for each atom, it indicates which molecule the atom corresponds to. For example, the first molecule will contain exactly 5 atoms. All of them are ones assigned because it's the first molecule. The second molecule will contain 4 different atoms, 2, 2, 2, 2, and so on. So that's the idea of molecular indicator. And then we do compiling the model. Once we define the model, we compile the model. I discussed it in previous slide. It's required for actually using the model by Keras. And finally, running the model. For this simple, very simple model, we use the fit method, which performs minimization of the loss. We discussed it in the previous slide. And if there are still questions about that, I can respond to after this slide. It's actually, I'm done now with this slide. Any questions?
So where is your label, like the 1 or 0? You say
the labels would be the output from first. The labels would be Y train. You see X train and Y train. The X train will be a set of features of all features of nodes and edges and pair indices in the input data. And Y train are the actual labels. So we pass X train the data, and Y train the labels. We pass it to the fit function. Train data set is a you see it's kind of produced from data and labels. Train data set includes both.
Okay, I'm new to this. Do you subset original data, or you put everything in and then find ones that, I guess you want to find the atom and the edge where that's causing it. Is that the goal for the exercise?
The goal for the exercise, as I said, is to show a simple example that give you some idea about the message passing network. And
Okay.
the simple which you can run yourself easily. And as for your question, I'm not sure whether I did not whether or not I responded already, but I showed you how the labels. So a Y train would be a vector of zeros and ones, labels for each number of, it's one-dimensional vector. The length of the vector would be the same as length of data. I have here a variable called ntrain, ntrain variable. Oops.
Yeah, I guess I get it. Okay.
Okay.
Thank you.
Okay. Any other questions?
I have a question. For the message passing step, where is code showing? So
You have a question about message passing, correct?
yeah, I feel like a message passing is kind of like a random walking, like a iteration from the network. Is that correct?
No, there is no, actually, it's not random working because there is no any probabilistic. It's not probabilistic, it's deterministic algorithm. Random walking would be probabilistic. You kind of generate a random number and then walk according to this number. But here, everything is deterministic. You have.
So this kind of just one step from random walking. You can iterate it like maybe 30 steps. But here, just one step from the first neighbor accumulated to so just
Right.
once, right?
Right. No, right.
Oh,
Right.
okay. So
you see, I just take adjacent nodes, whatever are adjacent, the nodes which will be used in computing the message and then computing the updated value of a given node. Those nodes are not picked at random. They are supposed to be immediate neighbors or one-hop neighbors of the current node.
Oh, okay. So the node get updated, and what's the edge updated?
I mentioned here the edges are not updated during the message passing. Edges are assumed to be fixed 0 or 1. And say if edge, for some reason, if you picked 0 edge, then the entire contribution containing this edge will be 0.
Oh, okay.
So we'll take only other contributions will be used when computing the message.
Okay, so
Okay.
back to the next slide, the current slide. So to my understanding, so the first message passing step is only updated for nodes. So all the other information didn't.
That's correct. That's correct. That's correct.
Okay. And then
So
next step convert it to the deep learning model. Right? You treat as the equally for the nodes, edge, and everything. Let's equally using a number 0, 1, something like that.
Well, 0, 1 is just example. Oh, well, yeah. Well, in other words, the message passing is doing basically a task similar to graph convolution, but does it in a more smart way. Graph convolution is also called filtering. It performs an update of the features of nodes. You can look at our previous class, which is available on the website, recording and the lecture slides, and graph convolution is discussed there. The message passing does very similar task of updating, but in a somewhat different way, which turns out to work better than just regular graph convolution. And I will support this argument later on because I have implementations of both graph convolution, a model with graph convolution and model with message passing. And you will see, you can run those myself, cartoon examples yourself, and you will see that the message passing network model produces more accurate results than the graph convolutional model.
Oh, okay, thank you so much.
You're welcome. No more questions about this one, I assume so. Or if you have questions, no? Then if you don't, you can ask later. I proceed to the next slide, which start discussion of the second prototype example of this class, the example that deals with molecules represented by sequences of tokens. And this example will allow us to introduce the self-attention network model. There would be inputs for this example would be of two kinds. First, it's a cartoon drug molecules represented by strings. Instead of smile strings for cartoon example, I will just use random strings randomly generated from a set of tokens, for example, A, B, C, D, E, F, G. And for example, fixed lengths, all the strings will be of fixed length, for example, 10. And a motive. A motive, it's kind of functional set of functional tokens. If, for example, ABC, if a drug molecule contains the motive, it will be considered good for treating a hypothetical disease and will be assigned a ground truth label 1. Otherwise, it will be assigned label 0 and will not be supposed to help in treating the disease. And the purpose of this example is to train a self-attention network model on this data so that it could predict the class labels for new, previously unseen cartoon drug molecules. The SAN model, SAN model is shown in the chart. Like in previous example, it comprises a set of tensors represented by rectangles, and the layers or transformations between the tensors. The most important one, which I mentioned here, all others which were discussed to some extent in the previous classes, and they are pretty much straightforward. I'll mention later on a little bit more elaborate on embedding layer, but for now, let's focus on the transformer layer. The transformer layer is a new layer we never discussed in the previous classes, and it's actually a hot area in deep learning right now. Particularly, it originated from natural language processing, but this construct, transformer construct, is used also in a number of other applications. The transformer layer is not actually a regular layer, but it is a composite construct comprising several regular layers, as shown here. And I will make two comments about the transformer layer. The first comment is that one of the layers, regular layers involved in the transformer, is called multi-head attention. This is actually the heart of the entire transformer layer or transformation. This is where the self-attention algorithm is implemented, and we will discuss that algorithm in more detail in the next slide. The second comment is about residual connection. This transformer layer involves two residual connections, this one and this one. The residual connection adds or sums up the input tensor, the input to a certain subnetwork, with the tensor which is output of the subnetwork. For example, this pink one would be added to this yellow one, which is output, and this plus sign means summation. And likewise, the light blue tensor here is added to the yellow, which is output from the subnetwork. The residual connection is a seminal technique first originated from image recognition, where it was shown to allow deep network to train easily and approach high accuracy. And this is discussed in the publication shown in the upper side. The entire network has been implemented as a separate executable, as one of cartoon executables, which will be available for you to run on your own yourself. I'll show you later on how to run executable. It's pretty straightforward. And you can see that this executable indeed does what it's supposed to do. In other words, it will predict the class label for new, previously unseen cartoon molecules. And the main conclusion from this slide is that in order to address the sequence analysis task, we do not necessarily need the recurrent layers or one-dimensional convolutional layers, which we previously in the previous classes discussed in connection to this kind of task. But this sequence analysis task can also be performed by the attention mechanism implemented in the transformer layer. And this conclusion is actually makes up a title for a separate, yet another seminal paper where the transformer was introduced for the first time. Its attention is all you need, 2017. Any questions? No question. Then I proceed to the next slide. And this slide introduces the algorithm, the self-attention algorithm, the algorithm implemented in the multi-head attention layer I mentioned in the previous slide. What this self-attention algorithm does, it allows to capture the long-range dependencies and relationships within input sequences and to identify and weigh the importance of different parts of the input sequence with weights which themselves depend on the input values. I want to stress that this would be dependent long-range dependencies as opposed to the message passing, which is local. It deals only with between adjacent nodes, but this is long-range. And in particular, one of the outcomes from this entire algorithm, which is shown in the flow chart, we will come to it later, would be the matrix of attention scores or attention weights. It's matrix A of size n by n, where n is the length of the input sequence. In other words, the elements of this matrix would be the values that indicate how important is each element to each other element in the sequence. Or in other words, how much attention is paid by each element of a sequence to each other element of the sequence. The flowchart shown here includes not only the self-attention algorithm itself, but also some pre-processing steps, such as, for example, we have original sequence of tokens. Then we break it into individual tokens. Then we replace the tokens by their order numbers. In this case, it would be just only 4 different types of tokens. So the order numbers will be 1, 2, 3, 3, 4. 3, 3 corresponds to C, and stuff like that. And then the final step of pre-processing is embedding. Okay, it's time to make a break. Let's do the break for 5 minutes. In my watch, it's 10:20 now. So we will meet again, continue in 10:25. I will start, we'll continue discussion of the self-attention algorithm. Okay? Okay, it's 10:25. We can continue. So I mentioned previously that the entire algorithm, the entire flowchart shown here presents not only the self-attention algorithm itself, but also some pre-processing steps. The last pre-processing step is embedding, where the sequence of sequence of order numbers of the tokens is embedded into multi-dimensional space of dimension D, which D may be a big number. And the purpose of embedding is actually to make different tokens represented by their order numbers equidistant one from another. So there is no any like, well, anyway, just embedding. And so after embedding, what we will have is a set of vectors of each vector is n vectors, and each vector corresponds to each token originally, and the dimension of the vector would be D, the dimension of the embedding space. And that tensor X would be the input, the entry point for the actual self-attention algorithm. The first step in the self-attention, the algorithm will imply a number of adjustable parameters. Those parameters are stored in 3 matrices designated WQ, WK, and WV, where the subscripts Q, K, and V are named after query, key, and value. The terms that are used in a web search, when you, for example, look for a video on YouTube, as described in this paragraph, you can read it. And I guess the entire algorithm was designed by analogy with the web search. But I will not don't want to go into details of that web search video. You can just read that information. And once we have those 3 matrices of adjustable parameters, the first step would be to compute the so-called projections. Projection matrices are designated Q, K, and V, and they're just multiplying. The Q, for example, is multiplying a result of multiplication of WQ matrix by the input tensor X. You get one matrix, another matrix, third matrix. The next step is computing the matrix of self-attention scores by using the so-called dot product attention dot product. In other words, the matrix A is you take Q, matrix Q, multiply by transposed matrix K, the superscript T stands for transposed, and divide by the square root, some kind of normalization, divide by square root of the dimension of the matrix K, the size of the matrix, I'm sorry, of the matrix WK, and then you take softmax. That's the algorithm for computing A. And A is the matrix of attention scores. It's one of the output from the entire algorithm. When you run the multi-head attention layer, it will do 2 outputs. One will be this matrix of attention scores, and the other matrix is matrix Z, which is kind of attention weighted version of X. It's the same as matrix X, but weighted by the attention scores. It is computed by multiplying the matrix A by transposed vector V, projection transposed matrix V, the projection matrix, and the size of n is n by dv. So the same number of n as a number of elements, original elements in the input sequence as in X, but the other dimension is different now. It's embedding dimension instead of the original D is now dv. Specified is just a hyperparameter specified by user. And the algorithm I just presented, outlined briefly, corresponds to multi-head self-attention with a single head. The term head, the concept of head is similar to the concept of number of channels or number of kernels in convolutional neural network. When we do multi-head attention, that means we basically repeat the same operation, but with different sets of adjustable parameters, with different matrices, Q, K, and WQ, WK, and WV. But in the particular implementation that I used for this example, in the biological example for this class, the matrix X was split into H similar matrices of the same size along the embedded dimension. So it is made assumption, the approach makes assumption that D, the embedded dimension, is a multiple of the number of heads. And then each head will be processed separately according to this algorithm, and the results will be concatenated at the output. And this approach, I found that it works better, produces better accuracy. This is why I use this kind of flavor implementation of the multi-head attention algorithm. Any questions?
So I have this question written in the chat. It's more like a basic thing. So in this example, you know ABC, you don't know, I presume we don't know ABC will indicating label equal to 1. You just want to find some motif, but you don't know the structure, the form of the motif in the ones, but you have the corresponding to the labels, right? So you try to see whether this data set give you some kind of consensus about some motif that will predicting the label equal to 1. Is that right?
Well, I'm not sure I understand the question, because the ABC is not the labels. These are data. We have to make distinction between the data and the labels. It's different. So the ABC sequence is a data X.
Is a motif you want to find, right? It's a motif. Is the motif you want to find, right? So you know, in a regular linear regression, you have X is like the motif.
Right.
Right.
The self-attention algorithm, it does not find motif. It's just one of several layers in the network. What is used to find the motif or detect the present, not find the motif, but detect the presence, whether motif is present or not, is this is performed by entire model. And this is only one layer in that model. So this layer itself does not detect motif.
But later, if you have new data come in, it's mostly for prediction, right? If you have new data come in, you'll go through this network, and then they will say, for certain molecules, your label will be 1. The predicting the label will be 1 for
That's
those,
correct.
right? So,
that's correct.
Okay, yeah, okay. I think I got it. Thank you.
Okay. Well, I will show you the entire model, which includes this layer in particular as one of steps. Oh, actually, I did already. I think yes, I did already in the previous slide. I showed the model. So once again.
Hi, can you talk about, you had mentioned in the last, before the question came on, you had talked about the reason why you chose a specific type of the flow or something like that. Can you repeat that again, that part?
You're talking about the next slide, correct?
Yeah, correct.
Okay, what type of flow you mean?
I think towards the end, you were mentioning that you found more accuracy with the specific
Oh, I
type.
see. Yeah. Well, when we separate the split the input tensor X into a number of subtensors, and we assume that the embedding dimension is a multiple of the number of heads that we use. I'll explain again, just repeat it once again, what I wanted to say. If it's not clear, you will correct me. So what I wanted to say is that we found then when we perform that splitting, the accuracy is better. The final accuracy of the entire model, not of this layer, but of the entire model, improves. alternative to this splitting, we might not split, just use original vector x with every time with number of heads h. With each head, it will be the same tensor x. Well, yes, it will be the same tensor x, but different sets of adjustable parameters. And hopefully we will get different results. But the problem with this approach that nobody guarantees that the result for different heads will actually, because the parameters, we do not set this adjustable parameter. They are adjusted automatically. And there is a way, there is a chance that actually multiple different heads of the algorithm will actually employ the same parameters eventually, because the parameters are adjustable on automatic basis. Whereas with this one, with this approach, it's guaranteed that you process each part of the input separately with, even though I don't know whether it's the same parameters or not, but it will be different processing because you use different data basically for each head. Okay,
thank you.
Does it explain
yeah,
the answer?
So basically, out of the full data, you are the sort of subsampling each head. Each blue is like a subsample of the input data that you are putting into tokens and repeating the formula again. That's right, right? You're subsampling the input X.
X is the same. Well, in all cases, whatever, multiple heads, X will be the same because X is just embedding of your original input vector. But then when you do embedding after embedding, you have multiple, for each element here, you have a vector of multiple components, decomponents. Those components are different. And you take a several subset of components of that embedded vector and process it in a separate, in one self-attention algorithm. Then you take another subset of components and process it differently. And the output, once you will have basically a set of matrices Z also. And then you will concatenate those matrices, sub-matrices Z to get the entire matrix Z.
Yeah, got it. Thank you.
Mm-hmm. Okay, if there is no other questions, I will proceed to the next slide. And this slide basically presents the algorithm for training the self-attention network. It's in a way similar to what we discussed for the previous prototype example. It includes the same basic steps of processing, which is header, defining a model, getting data, and finding, running the model. I only wanted to point to the location where we actually use the multi-head attention layer, which is will be here, layers, multi-head attention, and with some arguments, and the residual connection. We take, say, original, our input would be Y, then we do some processing with some subnetwork. We output a new value of Y1, and then we sum up Y with Y1. The Y1 would be output from the subnetwork, and we sum them up, and then we proceed further. And the same with another residual connection. Hope that is pretty straightforward, and you can look at it in more details. You will have this code available. You may ask me questions if you still have it, but I assume that's pretty much straightforward. Any questions, by the way? If not, I will proceed to the next slide. And actually, that would be start. First, before, okay, how to run those prototype examples. So you allocate an interactive session on BioWolf, then you load the model DLBio/class 7, which corresponds to the model that contains all of the prototype examples. There will be 3 prototype examples. The 2 main examples are shown in a solid box. This is MPN, mole graph classification, This message passing network applied to molecules represented by graph. And the other one is self-attention network applied to molecule represented by string. And in the middle, there is a auxiliary executable, which is similar to the first one, but instead of message passing algorithm, it implements the graph convolution algorithm. So by running these two examples, you can see that graph convolution produces much bigger error than the message passing network model. Instead of 0.2, you get 16, something like that. That's basically it. You can run those examples by just typing their names, and you will get result. I showed you in the slides only the training algorithm, but the actual executable that you will have available includes also the testing algorithm. So in addition to training data, there would be testing data will be generated, and which will not be a part of the training data set, and you will see on the testing data how accurate are the predictions from the model. Any questions? No. Okay, then we proceed to the second part of the class, the biological example. And the biological example is called MPSEN-MP, message passing and self-attention based networks for molecular property prediction. The code for this example is a composite code. It was running of this code is shown in our website. It's a separate application which you can load and then run. And it was the code base for this application was taken from 3 different repositories. One is Keras, and 2 others are GitHub repositories. One of the codes is originally developed in PyTorch, so it was partially re-implemented in Keras, and the others are both in Keras. The task of this application, basically, what it does, it takes molecules represented by either graph or a sequence of Smiles tokens, and predicts based on the data used, either a discrete property value or binary label, for example, indicating whether the drug is good or bad for treating a hypothetical disease, or it predicts a continuous property value or label. We also call it label, which indicates how good the drug is, for example. The key problem in applying deep learning to prediction of property values is that the amount of labeled ground truth data is limited. The labeled means the labels here are the property values, which are measured experimentally. And this is a laborious and expensive operation. So the amount of training data typically is limited. As a consequence, the model training the model may result in overfitting. We discussed overfitting in previous classes. I'll briefly refresh your knowledge about it. Overfitting occurs, it's a phenomenon that occurs when the amount of training data is tiny, very small, and the model is very complex. It has a lot of adjustable parameters, so the model fits this particular training data set too well, and it focuses entirely on this small training data set, and it is unable to generalize the training results. In other words, to make accurate predictions for data that were not a part of this training data set. And obviously, there are 2 general approaches to fight overfitting. One approach is to increase the amount of training data, if you can. The other approach is to reduce the complexity of the model, reduce the number of adjustable parameters in the model. And in this biological example, we'll explore 2 techniques to address the issue of overfitting. The first example would be to increase the size of training data set by doing data augmentation. Data augmentation means we will, for SMILES, we will consider multiple alternative SMILES representations of the same molecule. Of course, they will correspond to the same property value, but you will effectively increase, by augmenting the data, you will increase the effective size of the training data set. And hopefully that will be sufficient for more accurate predictions. And it is indeed sufficient, as we will see later. This is a good, it's our preferable approach. The other approach that will be tried is to decrease the complexity of the model and decrease the number of adjustable parameters. And for that purpose, we will use a technique termed transfer learning, where there are actually two models will be discussed instead of one. And those models share a certain block of parameters, of layers together with adjustable parameters. And somehow we will be able, using this approach, to reduce the effective number of adjustable parameters in the model. And details will be understood later, will be presented later. Any questions?
Hey, I have one question. Could you, an alternative here, because the problem is that SMILES is ambiguous. What if you just picked an unambiguous way of representing the data?
It is ambiguous, that's true. But I'm running a little bit ahead. I'm going to discuss it later, but I will give you a quick answer that ambiguity is not a problem for the message passing network model, because however many SMILES representation you able to generate, the graph is unique, and the message passing network model employs graph. It does not employ the SMILES strings immediately. That's a quick answer.
Okay, thanks.
Okay. Any other questions? Okay. Later on, if you have questions, let me know. And I proceed to the next slide, which presents basically gives an overview of the algorithm, of the training algorithm used by our application. Shown in this slide is not the entire algorithm, it's certainly much more sophisticated, shown only the main function of the training code of this application. And the good news, which I already mentioned, that was true for previous classes as well, that the main function, which actually governs execution of the entire code, comprises the same basic processing steps as we did for prototype examples earlier and for all other examples in this course. These are header, getting data, defining models, and finding the model, running the models. The header in this case will be just kind of pre-processing of parameters of the model. There is also import statements, of course. They are not shown here. They will be later upper, and I'm showing at the very bottom of the code. And there are, of course, import statements, but they're not part of the main function. Then there will be getting data. In this case, just get data with options, command line options. You get training data, validation data, and so on. In the getting data, we have several items when we discuss that, several items that will be discussed in particular. These are the data sets that will be used by our example. The data sets names shown in red font are new. They were not used in previous class. This is why the current application employs an extended set of data used by class number 5. Then all other, the black ones shown in black font, they are the same. They were used previously. Then the step of defining model, we will discuss message pass and self-attention, a little bit more elaborate on message pass and self-attention. Then BERT model and send BERT model. The term BERT comes from bidirectional encoder representation from transformers. It's some kind of advanced techniques that come from the natural language processing, and I'll give you more details about these techniques. And finally, running the model, actually there are not a single executable. Well, executable is the same, but training the model can be performed in 3 different modes. The mode may be dependent on the model that you use and depending on the data that you use. You may either train mode, run in train mode, or pre-train or fine-tune. These are all different modes of training. And the entire code comprises 4 executables shown in green font. The preprocess executable, train, predict, and visualize. The preprocessing executable, I'll give a little bit more detail what it does, but actually it's optional because all the data are already preprocessed, and it's optional if you want to preprocess yourself. The training executable will actually perform the training, as I said, in one of 3 modes. Then prediction will predict the labels, the continuous labels for or binary labels depending on the data. And finally, visualization will visualize the results for some of the data sets, and example will be shown later in this class. Any questions? Okay, I proceed to the next slide for now, which will discuss the data used by the MP-SUN MP application. The entire data set comprises 6 sets of data, and they are naturally split into 3 couples. The first couple of data in the left column are the data with continuous property values. This is JEC2 and log-P. Those are data that were already discussed in class number 5, the same data. The other set of data couple are the data with discrete property values, these binary labels. The first one, let's a little bit more elaborate a little bit more about the first 2 data sets. So JEC2 is a data set relatively small, approximately 2,000 entries. The property value will be the Janus protein kinase 2 inhibition coefficient. So some kinetic rate constant is property value. The second data set, log-P, is the property value will be the logarithm of the n-octanol water partition coefficient. For biologically active molecules, it is known to have higher value than for any other molecules. For next couples of data, the BBPP. Oh, by the way, each data set comes in 2 flavors. One, the original data set, and the other is augmented. So this suffix A stands for augmented data set. BBBP is a new data set we did not discuss previously. It's a blood-brain barrier membrane permeability. So if a certain membrane between blood and brain is permeable to the substance, the label will be 1. Otherwise, the label will be 0. And the B-log-P is the same as log-P, but it's a kind of binarized version of the log-P data set. Instead of continuous property value, we use binary label. If the continuous property value is above a certain threshold, the label will be 1. Otherwise, the label will be 0. The reason why I consider this binarized version of the log-P dataset is that this exactly, or approximately, this dataset was used in one of publications, and it would be interesting to compare our results to the results reported in that publication. So the right column of data sets are the data without any property values at all. These are Campbell and zinc. Campbell is a data set from chemical database of chemicals of European Molecular Biology Laboratory, version 33 of the database. So it just reports the SMILE string and the Campbell ID. And there are the other, actually every data set reports 4 columns. That's a result of pre-processing. For each molecule, we report the number of atoms in that molecule and the number of tokens when we break the SMILE string into tokens. For some reason, that is used by the model. This information is used by the model. And finally, zinc is a random subset of randomly generated subset of size 25 million out of several hundred million of molecules or SMILE strings from the zinc database. Zinc is a database of commercially available substances with biologically active properties. Each couple of data sets comprises one smaller data set and one bigger, approximately an order of magnitude bigger data set, and that allows us to explore the effect of the size of data set. Okay, and I mentioned that for data set with property values, we also consider the augmented version. Augmentation approximately 10-fold augmentation. So for each SMILE, there would be approximately 10 different alternative SMILEs strings would be generated by augmentation. Any questions? Okay, I proceed to the next slide, which will be actually this slide, and the next slide will be will report the most important result of this entire class. The result will be that the message pattern network using the augmented data dramatically overperforms the self-attention network result in prediction of as indicated by these cells in the table highlighted green. I will proceed discuss this table later on, but let me first start from discussion of the models which we compare. So we compare in this slide message pattern network model and self-attention network model. They are similar to the models we already discussed in the introductory part. The only item that needs to be commented is the pre-processing, because this model use real SMILEs data as input, as opposed to cartoon data we used in the prototype examples. So in both cases, 2 models, the very initial input is SMILEs, SMILE strings, a set of SMILE strings. But these inputs are pre-processed in a different way by 2 different models. In the case of message pattern network model, the pre-processing program is called featurizer. The featurizer program makes use of expert knowledge about molecules, and it implements that expert knowledge in the input tensor, input graph, which is generated from the SMILEs. So for example, if you have in the SMILEs say token O, oxygen, it's not just token, but we know that oxygen has valence 2. Then we look at adjacent atoms, which also we know the valences of those atoms as well. That's a kind of expert knowledge. Then based on this knowledge, we can figure out what would be the bonds between these atoms, which is single bond, double bond, triple bond, aromatic bond, and we can figure out number of hydrogen ions attached to these heavy atoms, like atoms like oxygen or carbon or whatever, nitrogen. And this is a kind of process, and eventually we come to the atom features matrices, which I mentioned previously, will be some matrix of zeros and ones, the bond features matrix, the pair indices matrix, and molecular indicator. With self-attention based network model, the pre-processing is termed tokenizer. The tokenizer is just breaking the SMILES strings into tokens. And those tokens are not necessarily the atom symbols. It may be opening parentheses, closing parentheses, equal sign, and many other possibilities. I'll show a little bit later on examples of what we will discuss, consider as tokens. So it's much simpler at the output, at the input, much simpler pre-processing. But the relationship, long-range relationship between different tokens, between different elements in the input will be determined later on when we apply the transformer layer or self-attention algorithm. And then we will identify the long-range relationship between these tokens. Now I proceed to the discussion of the table of the results. The table reports, there are two kind of inputs, output, I mean, two kind of data in the table. The first for the JAK2 and log-P dataset, as well as augmented JAK2 and augmented log-P, where the property values are continuous property values. We report the mean squared error between the predicted property values and observed property values. Predicted is what we train the model on the training data, then we take testing data set and we run the model already with adjusted parameters. On the testing data set, we predict a set of property values. But for the testing data set, we know the true values, the ground truth values. And so we compare the two sets of property values. And the mean squared error between the two sets of property values, the predicted and observed, will be reported in the table. For the second set of data sets, which is the data sets with binary property values, binary labels, the reported value will be percentage of incorrectly predicted binary labels. So now I looked at the table itself, message passing network and self-attention network. You see for the original JAK2 dataset, not augmented, the numbers are comparable. But if we use augmented data, the message passing network model produces much more accurate result, much smaller mean squared error than the self-attention network. And the same repeats for all the data sets. You see, consistently, when we use message passing network model with augmented data, it consistently produces much more accurate results than the self-attention network. And according to, I looked at the literature, it looks like that result was, that phenomenon was not known. So it has been reported for the first time in this class. Any questions about this slide or previous slides?
Can you remind me what's data augmentation? What do you exactly do?
You ask about how much augmentation. I said 10x augmentation approximately. So if for a given molecule, it's big enough molecule, you basically may generate many different alternative SMILES version, but you pick only 10. However, if molecule is not that big, you will take whatever number is generated, is total number of the alternative SMILES is less than 10, we just take what is available. So it's only roughly 10x is only roughly the number of folds in which you increase the size of the data set by doing augmentation. Any other questions? Okay, now I proceed to the next slide, which the purpose of the next slide is to elaborate a little bit the results of the previous slide. It's also a very important slide, and it focuses on one of the data sets we discussed in the previous slide, the JEC2 data set. So the chart shown here compare the predicted versus observed quality values for the message pattern network model shown in red dots and the self-attention network model shown in blue dots. And the left chart corresponds to the original non-augmented data, whereas the right chart is generated using augmented data, approximately 10x augmented. So when we compare observed and predicted quality property values, the ideal prediction will correspond to the data points located exactly on the diagonal, because here the predicted is exactly the same as observed. And you see from the right chart that the self-attention network model, the blue dots, it's about comparable to what we had for unaugmented data. But the message pattern network model on augmented data makes much more accurate predictions. Almost all of the data points are on the diagonal or close to diagonal. And the numbers shown here indicate that it's multifold improvement of the mean squared error for message pattern network as compared to the self-attention network. Oops. So I wanted to give my understanding, my interpretation of these results. Why the message pattern network succeeds and the self-attention network does not succeed on augmented data. With self-attention network, we are dealing with the part of this model employs the self-attention algorithm, which finds the relationship between different elements of an input sequence. And this in particular takes into account the location, the position of the sequence, of the element in the sequence. But when we do augmentation, the augmentation basically reshuffles all the atoms in all the tokens in the input. And that reshuffling, so we, after augmentation, we come with a completely different sequence of tokens. And that reshuffling may confuse the model. And that's why kind of, on one hand, there are two kind of trends. On one hand, you have more data, but on the other hand, the model may be confused by this reshuffling. So the results may not be, they may be a little bit, and if you can see, look at the table, there is some improvement, a little improvement on augmented data for the self-attention based model, but it is not significant. As opposed to the message pattern network model, for message pattern network model, which uses the graph as input, it doesn't matter whether it's reshuffled or not reshuffled, the smile string reshuffled or not. The graph is the same. The graph is unique. And this is why the message pattern network only succeeds because it takes advantage of the amount of data, but does not suffer from the reshuffling. And it, as a consequence, it produces much more accurate results. Any questions? Okay, let's proceed to the next slide. We have seen the success of the message passing network on augmented data. Now, a natural question comes up. Can we do better with self-attention based model as well? And the approach suitable for addressing this question is termed transfer learning. In the transfer learning, what transfer learning is roughly is when knowledge learned from a task is reused in order to boost performance of a related task. For the particular problem at hand, the problem of molecular property prediction, this approach was implemented in the application termed SmilesBird. It is published, and there is also a GitHub repository of code. And according to this approach, the approach takes advantage of the analogy between processing of sentences and words in the natural language processing and processing of smile strings and individual tokens in the Smiles dataset. The general idea of the approach is that instead of a single model, let's consider two different models, the source model and the target model. The source model solves somewhat auxiliary task. That task does not require ground truth labels. I will describe the task a little bit later. It does not require ground truth labels, and therefore there is a lot of a vast amount of data that we can use to train the source model. Then once this training is done, we use train on the target model. The source model and the target model share a certain set of layers, a block, which in this setting is called BERT encoder. BERT stands for bidirectional encoder representation from transformers. It's just some concept that came directly from natural language processing. So the BERT encoder block is common to these two models. And when we do training of the source model, we adjust parameters of the BERT encoder. Then after this step is called pre-training. The second step is called fine-tuning. Here we train the target model. Now, when we train the target model, the parameters which we learned from the pre-training step now are frozen. They use those parameters that we found already. They are frozen, and that entire block is used as a part of the target model that is trained on the target dataset, the same tiny small dataset, but because significant portion of the parameters is frozen already, the remaining parameters, it's not so many remaining parameters, and hopefully the reduction in the number of adjustable parameters will address the problem of overfitting. And this approach was used by the SmilesBird with some success. So the SmilesBird probably can be regarded as a current state of art in applying transfer learning to molecular property prediction. So it's why it's worth considering this approach and comparing it with other approach. So one more comment about the pre-training stage. I said it's a different task which does not require ground truth labels. What is done at the pre-training stage? We take Well, actually, it's the same. Will be explained on the same next slide in more detail. But basically, the at the pre-training stage, the input would be a smile string, and the output would be also a smile string of the same length, of the same size. But the input, we purposely mutate the smile string by making some tokens at random, or substituting some tokens with other tokens. And we train the model to recover the mutated tokens at the output. And this way, by running this pre-training procedure, we expect to determine the set of parameters of the BERT encoder. Any questions? I proceed to the next slide then, which actually shows the model, the source model used at the pre-training step. It takes as input a smile string, which is at the pre-processing step is mutated, and we have a sequence of the mutated token order numbers as an input. Then we perform embedding, and at the output we show the original unmutated sequences of tokens, order numbers, sorry. And the entire model comprises the BERT encoder, as I already commented, and then the remaining part of the model is termed classifier. It involves two dense layers plus some other layers, which does not involve adjustable parameters. So the dense layers basically are the only source of adjustable parameters in the classifier. But of course, all the parameters of BERT encoder will be used as well in the training. And the BERT encoder is just a set of consecutive transformer layers. In this implementation, it was 6 by default transformer layers. We already discussed the transformer layers. So this model originally was implemented in PyTorch, and SmilesBERT is PyTorch. For this class specifically, it was re-implemented in Keras, and the re-implemented version I named SandBERT source model. It is similar to SmilesBERT, but implemented in Keras. I already mentioned BERT encoder involves 6 consecutive transformer layers. The BERT classifier, 2 dense layers plus some other layers without adjustable parameters. The inputs and outputs to the source model would be the sequences of tokens, the sequences of fixed lengths specified by a user. By default, it's 64. If the actual smile sequence longer than 64, it will be discarded. But you can change, you can increase the maximum length, allowed maximum length of a sequence. If the actual sequence is shorter than 64, then it will be padded by adding the pad control symbol. And the other, this is a set, by the way, example of the tokens that are used by the program. The total number of tokens 110, and it includes also a couple other control tokens. One is unknown. If, for example, we detect tokens which is not a part of our vocabulary, a token, like we process new data, and there are some token that usually typically it's rare token. If it's not a part of our vocabulary, then it will be replaced by the control token unknown. And the mask token, also control token, is used for masking of the input. Masking means replacing of the actual smiles token by the mask token. But at the output, you try to recover the original non-mutated sequence. And the results of pre-training are reported in the table. There's no much to say about this one. Just report the final score, final loss were obtained with two different data sets. The number of epochs that was used in pre-training was 10 epochs for zinc data set, because it was the number of epochs reported in the Smiles Bird paper. And I tried to follow that recommendation. And for the Campbell dataset, which is much smaller, I performed 30 epochs and got somewhat better accuracy of smaller loss and better accuracy of training. Any questions? No questions. And I proceed to the next slide, which reports the results of fine-tuning and the prediction of the property values using the Sunbird target model. The target model is similar to the model shown in the previous slide. The input, but with several differences, the input will not be mutated. It's just a regional input sequence of order numbers generated from Smiles. The bird encoder block used here will be employ the frozen parameters obtained from the pre-training stage. And instead of classifier of the previous model, we use fine-tuner, which also has two dense layers plus some other layers which are not shown here with without ingestible parameters. And the fine-tuner outputs a scalar, which will be the scalar will contain the property value that we computed. The results are shown here. The results are not that impressive as we have seen for the message passing network model. We see that the SAN model and then Sunbird model, three models basically. Sunbird model pre-trained on Campbell data and Sunbird pre-trained on Zinc data. We see that only in some of the cases, the transfer learning approach allows to improve the reduce the accuracy. Improve the accuracy, reduce the error as compared to the original SAN model. And even when this improvement is found, the improvement is not dramatic, as we have seen with the message passing network model on augmented data. And for one particular cell of the table, this one, the similar value was computed by the Smiles Bird in the Smiles Bird paper. It was reported their number was 8.5%. It's comparable with our number, but the main point is that both the numbers are much bigger than the number we found for the same data set using message passing network model, which was 0.05%, much better. So the main conclusion from this slide is that the message passing network model with enhanced by augmentation remains the winner out of all the models discussed in this presentation. The second conclusion, other two conclusions are regarding why we do not succeed with the transfer learning approach for this task. To succeed, but it's not as good as for message passing network. It's only partial success. The main reason why it doesn't work, it's not because the transfer learning approach itself does not good, but in the transfer learning, we say that we want to boost performance of a related task. Given a knowledge from a given task, we want to boost performance of a related task. And the notion of a related task is not strictly defined. There is no rule. This task is related, other is not. We just try and see what happens. And for this particular setting, I think that the task solved at the pre-training, it's called mixed language modeling, when we mutate some part of sequence and output, try to recover, it's kind of very different task from the prediction task. So our pre-training does not provide optimal performance as a prediction step because it was focused on different tasks. And that basically what the reason, and I see it actually in the actual fine-tuning when I run this code, the loss or validation loss does not reduces a little bit and then stop reducing beyond a certain value. That's because there is some constraint imposed by the training, pre-training, that does not allow the fine-tuning step to perform well. That's basically my understanding of this not so impressive result. Any questions?
Yeah, I have a question regarding the values, the results. Are you able to hear me? Sorry.
The question about the data?
Yes. So the table that you've shown here. So when do we consider a value to be good, and when do we say like above the threshold where we say that the values are?
Oh, I see.
impressive?
I see.
And
I see
also,
what you
how
mean.
do you interpret that? Yeah.
Okay. So the threshold 185, 88, this one, right?
Yes. Okay.
Okay, so that actually was the threshold that was used in the Smiles Bird paper.
Oh no, I'm sorry. What I was trying to ask was that, like, the table that you've shown, where you compared the results from the different models. How do you determine if a model worked well, and if how do you? Because I think we can agree that one of the results was not impressive. So how do you determine if the result is good and acceptable? Right now, based on those values.
Okay, it depends. It depends on the particular data set. We look at, if we look at that, we report in this table, we report the error. And the meaning of the error is different depending on the type of data. In one data set, it's mean squared error between predicted and observed. And we just compare the number, compare it with other numbers for other settings. For example, this number is compared to the message passing network, the similar number, but generated on unaugmented data set. And similar numbers generated using message self-attention based model. And we compare the numbers. The smaller the number, the better. It's obvious, right? Because it's error. The meaning of these numbers are error. In one case, it's mean squared error between predicted and observed. In another case, it's percent of error in prediction of binary labels. In other words, the percentage of incorrectly predicted binary labels. Obviously, that the smaller this number, the better. And you see this number is the smallest one out of all other models, all other data sets. It's pretty clear. I think it's pretty straightforward. Does this address your question?
Yes.
Okay, and then similar, basically we report similar number on the last, in the last table, but I did not consider the augmented data on that slide, because the purpose was to not to evaluate the augmentation effect, but to evaluate the transfer learning approach, how transfer learning without augmentation helps to improve the accuracy of predictions. So you see these numbers are if they are green, if they are better, smaller than original, but not dramatically smaller. The most successful is BBP. It reports improvement for both Campbell and Zinc source data, and twofold for Campbell, twofold improvement, which is good, of course, but it's not comparable to what we see for message passing network model. So the message passing network remains the clear winner out of all the models discussed in this presentation. Any other questions?
Yeah, follow up on that prediction error. So in your original data, is the proportion of prediction, like how many were labeled, the proportion of their label as 1 affecting your performance? Is, you know, like if I'm giving a new data set, I have no idea how to, you know, like what the cutoff is. It 50%? About, if my prediction accuracy is about 50, maybe towards 70, and you consider, oh, this model, the data have something information, and then the network is doing its job and then have good prediction. So that's just like a rule of thumb. How do I determine?
You determine by using the testing data set, which is kind of evaluation and sometimes testing step or evaluation step. You use the data that were not a part of training data set. That's the most important point. It should not be used for training. It's just completely independent data, which you just may say, if you want, you have your own data set, you may want to split your data set into the training part and testing part, or maybe also validation part. It depends on how you want to proceed. And you train the model only on the training part of data. But when you do this evaluation, where I compare, I do not use the training data set. I use only the weights, the adjustable parameters I learned from training. And then I apply the model with this adjustable parameters, already fixed parameters, to the evaluation or testing data set, which was not a part of training. Does this address your question?
Yes.
You can do the same with your data, of course. You can. Any other questions? Okay, then okay, I guess we skipped the second break. Let's finish then with the presentation, and then we'll make a break because we missed the time already. So this slide just explains how you can run the MP-SAN MP application on Bible. The more details can be found in this user manual page, but this is a very brief outline of the procedure. You allocate an interactive session, then you load the model MP-SAN MP. Then you, if you look at this environment variable, it points to the set of available the source code, actually, as SARC, MP-SAN MP SARC. It's all the source code which is used in the application. Then you have MP-SAN MP data folder, which stores all the data that you may want to download to your local folder. Then you run the train executable with appropriate command line options. Depending on those options, the option in particular specifies the model that you use, MPN, SAN, or SAN-BERT, and the data that you use. It may be BBP, JAK2, log, Campbell, Zinc. Depending on the model and the data, the code will figure out which mode you are running. For example, with this set of options, it will be training mode. With this set of options, because the data sets are Campbell or Zinc, both without ground truth labels, it will be pre-training mode. And the last one, it will be fine-tuning mode. But it's the same executable. But depending on command line options, it will do a different processing. Then once you have done training, the results will be stored in checkpoint file. And then you can run a predict code, which runs on testing dataset, different from training dataset. And you specify as one of inputs the checkpoint file, which contains the trained adjusted parameters, adjusted during training step, and you generate some output, the prediction results, and then you can visualize the results by running the R executable, visualize R with following this command line. Okay, you can see get more details by looking at this user manual page, and this final slide is summarizes what was presented in the this class and make some conclusions. So in the introductory part, we discussed the message passing mechanism and self-attention mechanism. We mentioned that the message passing layer employs both node features and edge features, as opposed to the graph convolution layer. So the message passing is kind of generalization of the graph convolutional layer. The message passing layer, like a graph convolution, is still local transformation of data, as opposed to the self-attention algorithm, which is long range, captures the long range relationships. The matrix of attention scores, it shows the attention of each element, even distant element in the sequence, the relationship between distant elements in the sequence. And in the biological examples, we discussed the models that use both of these models and different types of data, either represented by either graphs or smile strings. We discussed two different kinds of tasks solved by the model: the classification task for binary labels, discrete property values, and regression task for continuous property values. We discussed the problem of overfitting and how we handle this problem with two approaches: the data augmentation, using data augmentation, which works well for message passing network model, but not so well for self-attention based model, by the reason that I mentioned previously. And to some extent, the overfitting maybe in some cases, it depends on the data, may be addressed also by the self-attention Sandberg model using transfer learning. And so as a result of this discussion, I certainly can say that the message passing network model is preferable. It's a clear winner out of the models we discussed when it is used with data augmented by SMILES enumeration. And the SMILES transfer learning may or may not succeed, depending on the source task, which is supposed to be related to the target task, but it's not strictly defined issue, so it you may or may not succeed. Any question? That's it. The entire presentation. That's it. We can now make a break, and then if you have additional questions or discussions, or you may also try to run the your software yourself. I can give you a little bit more comments about the user manual page presenting this application. Any quick questions so far?
So about this is more like a black box approach. At the end, I will have a prediction on a new data set. But if I want to see what's the key part, is there any way we can look into the black box and tease out some major part that was determined? Like, for example, if we want to find a motif,
Okay.
like, is there any idea, give me any idea about what the motif would look like that determine the prediction?
Okay, actually, I think if you want to find the motive, probably you want to use a different application, a different program This particular application and this class focuses not on finding the motif, but on predicting property value. So it basically will tell you whether or not a motif is present, if it is motif at all, because I use motif as a kind of cartoon example. In real data, it may not be a fixed motif, as I showed, contiguous set of tokens, but it might be multiple different even distant tokens located within a sequence. So, I mean, the quick answer to your question is that this particular application does not perform prediction of how the motif looks like. You need a different application. For example, one possibility, you may try maybe not exactly, but class number 2 discusses in more details application of more close, I think, to the question that class number 2 attempts to address is more close to your question.
Thank you.
You're welcome. Any other questions?
I have a question regarding the augmentation. So, in this, the augmentation was possible because of the structures of the smiles, and for every structure there are multiple potential smiles. But so is this augmentation specific to these kind of problems, or are there, or can we apply this augmentation in all, in even other types of problems?
No, of course you can use for other data. Augmentation is implemented in a separate package. Let me see. I listed it somewhere, but I did not mention. Yes, it's here. Smiles enumeration. It's application available on GitHub, and I use that one for augmenting the data. It's implemented as a part of the pre-processing. We have seen that there is a pre-processing executable. The pre-process executable, it employs the code from that smiles enumeration application, the code, and it performs augmentation for you. You pass to this executable. The usage of this executable is explained in the user manual page for our application. You can see how that can be done. Besides, the code is available if you want to look how exactly it was implemented in our code. You can look at sarc, ampersand sarc, and you see all the executable, all the source code that makes up the entire application. You can look at pre-process executable, and you can see how we do it. And in principle, yes, you can, maybe with some minor modification of the code, you can use, perform augmentation on your own data. I think that's the answer to your question. Is it? Is it
Yes, I think
good enough? Yes, it may require some minor modification, but you will see the idea. If you look at this code, you will see the idea how we perform augmentation.
thank you.
Any other questions?